Alright, so in these notes you're getting a very basic overview of replication. And we'll talk about transcription and translation later, but these notes are replication. So in replication, to replicate means to copy. And your cells need to copy your DNA before they can divide. So one cell divides and makes two cells. This is how you replace cells if you ever got injured. This is also how you grow. Sometimes cells just get old and they need to be replaced. So if you're ever going to go and make a new cell, you have to copy your DNA because every cell with a nucleus in it needs an identical copy of DNA. And any cell that gets a messed up copy of DNA is going to be destroyed. If you pass on defective DNA, you have enzymes that will go and destroy those cells. So we need a perfect copy of DNA passed on before division. So the DNA molecule is two complementary strands. So they're not identical strands. So here you have a C on one side, and then you have a G on the other, and then you have an A on this side, and you have a T on this side. So the two halves of the DNA are not identical. They are complementary. And so you need to separate these strands, and you need to build complements of the original. And so the gray ones that you see here are the new strands that are being built on using the old strand as like a template for that. And then you'll end up with two identical pieces of DNA in the end. Each new DNA molecule is half old and half new. So if the blue is old, then you can see here that the gray is what's being built, and that is new. And so at the end, you'll have half blue and half gray. This is called a semi-conservative process. Semi means half, and conservative means keeping things the same without changing them. So we're sort of keeping things the same. The strand itself, when you copy it, it's not identical to the original because that DNA, the blue stuff, was the older stuff and the gray stuff is the newer stuff. So it's not 100% identical, but the way of splitting your DNA and using that as a template to build two new strands that are half old and half new is a semi-conservative process that also allows you to make fewer mistakes. And again, it's really important to not make any mistakes in your DNA because then you could build proteins incorrectly, which if you got a bad mutation could end up killing you. So this is a very important process. This is called DNA replication. It happens during the S phase of interphase. So you have a cell cycle. I think you have a picture in your notes of a circle, and you have these cycles of the cell, and you have S phase, G1, G2, and M, and these are just like what your cell does. So it starts with G1 and it grows, and then eventually it gets to the point where it's big enough that it needs to divide. But before you can divide, you need to copy your DNA. So S stands for synthesis, because you're going to make some DNA. And then after division, or then you go through another growth phase where you get ready to do mitosis. You copy the organelles, you get everything ready to go, and then you go through the M phase, which is then mitosis, where you split the cell into two cells. So a semi-conservative replication, like I said, it means the DNA molecule is half old and half new. Like here, if blue is old, in the end you end up with strands that are half blue, half gray. So they are identical to the original. You can see here ATCG, ATCG on both strands. It is the same, but they are half old and half new. So semi-conservative, we're not putting the blue strands and the gray strands back together. We are splitting it and doing half blue and half gray. DNA is very large. It would take you years if you started at one end of a organism like yourself, who has about 6 feet of DNA in each of your cells. If you started at one end and you just went down, all 6 feet, it would take you about 50 years to get to the other end. You don't have 50 years. Some cells in your body need to be copied every single day. Like skin. You get a new layer of skin every single day, which means that those cells in your skin need to be copying themselves constantly. And so you need to speed that up. You don't have 50 years. You've got about 24 hours. So to make this go faster in larger organisms like eukaryotes, you don't copy from left to right, one end to the other end. You open up thousands of replication sites, they're called bubbles, at the same time. And you read within that bubble, and then you just glue everything together at the end. This does not happen in bacteria, who have much smaller genomes and are much easier to copy, and they just go from left to right. But in eukaryotes, more complex organisms like you do need to go into these thousands of bubbles. Here's a picture of that. You're supposed to label this picture in your notes. You have an origin of replication. We're going to start here. You open up a bubble, you read to the left, you read to the right, until you reach the bubbles joined together. So if we read until we run into the next bubble, then those cell machinery are done. And then you glue these gray pieces together at the end. So the blue one is the parent strand, it's original. The gray one is the daughter strand, that is the copy. And then in the end you end up with two daughter DNA molecules that are semi-conserved because they are half old, the blue, and half new, the gray. DNA replication has a lot of enzymes, a lot, and you'll see more of them later in the week. These notes are just basic overview of replication. Two of the major important enzymes are helicase. Helicase is the one that unwinds the DNA. So this would be helicase right here, this green one, and it's going to go through and it's going to untwist that DNA because you can't read the twisted DNA. It also has to be separated because it's got those hydrogen bonds. You need to break those, so helicase does that. And then behind helicase comes DNA polymerase. Later we will talk about an RNA polymerase, and they have different jobs. But this one is DNA polymerase, and it will use the now unwound, separated, unbonded strand as a template, and it will sit on that and it will then add nucleotides according to base pairing rules. So if the polymerase, which is this big green one here, is sitting on a DNA strand and it sees an A, it knows that it needs to grab a T floating out in the cell. If it sees a G, then it knows that it needs to grab a C that's floating out in the cell and bond that. And it sounds kind of slow, but they can do this really fast, like thousands of base pairs every minute. They move very quickly, even though it sounds like it would be kind of slow. They're actually really good at this.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So there's actually a little bit more here to your notes. So after you're done with this process and you've made 2 strands of DNA, they're not perfect. Your DNA polymerase makes mistakes. So like here in this strand you have A, T, C, G, that's all great, and then DNA polymerase added a G here to a T, and those don't match. That's a mismatched pair. And so this should be an A. And that error most of the time is nothing. So most of the time when you get a mutation in your DNA, it doesn't code for anything, it doesn't change anything, it's not that big of a deal. Occasionally you'll get a mutation in your DNA which is negative, and that could lead to something like cancer, or you could get a change in your DNA and actually give you something cool, like maybe it gives you longer fingers, or it gives you a different skin color or eye color than was available before. And so this is, that's what happens in mutations. So DNA polymerase can go back and reread the DNA, and so every 1 in about 100,000 base pairs is wrong, and they make mistakes, and so they can fix them. And so DNA polymerase can then proofread the DNA, and after it does its proofreading process, then the error rate is now 1 in 1 billion. So the DNA polymerase reads the DNA, and where it sees letters that are wrong, it can replace them with new letters, and so that leads to a much smaller error rate than 1 in 100,000. So it doesn't fix it perfectly. Your DNA is never copied perfectly, but the error rate is pretty low. And then, like I said, most of the DNA damage that you get in your life, either from copying errors, which most of your damage is copying errors, or from chemicals, or from sun exposure, most of that does nothing to you. Sometimes you can get a negative mutation like cancer, and sometimes you can get a positive mutation. Like in some humans, like 7,000 years ago, there was a mutation that allowed humans to be able to drink milk as adults, and that provided a new food source for adult humans that isn't available to the majority of humans. About 65% of humans still cannot drink cow's milk today. Here in this picture you have 2 birds. If a mutation gives the bird a longer beak, and they use that to get insects hiding in trees, then that benefits this bird, and then that bird is more likely to reproduce, have kids of its own, survive, pass on the longer beak to its offspring, and that's how mutations persist. They tend to have more kids if the mutation that you got is beneficial.
The DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high-frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and billions of times a day.
So the last part of this assignment is this coloring page, and this is similar to what you did last week. So you're going to color your bases a certain color. When we read through here, it says each time a new cell is made, the cell must receive an exact copy of the parent cell DNA. The new cells then receive the instructions and information needed to function. The process of copying DNA is called replication. Replication occurs in a unique way. Instead of copying a completely new strand of DNA, the process saves or conserves one of the original strand. For this reason, replication is called semi-conservative. When the DNA is ready to copy, the molecule unzips itself and the new nucleotides are added to each side. Note the nucleotides are shown in their three parts. You're going to color the sugar blue, the phosphate pink, and the four bases, and that's here in the box. Notice that several nucleotides are floating around. They're waiting to pair up with their match. The box section shows two new strands of DNA. Color the old strand and its included base red, and the new strand and its included base green. So first, go to the drawing tool. You're going to change your transparency. Make sure you turn that up a bit so that we don't color over the lines. And then let's start with pink. So all these circles here, these are phosphates. So you're going to color all the circles in pink. So everywhere you see a circle, you're going to color a phosphate. Then the sugars are pentose sugars. So in this case, it's deoxyribose. And you are going to color the deoxyribose blue, light blue, dark blue. I don't think it matters. Nope. Let's go with light blue. So you're going to color in all of these pentose sugars here. This is deoxyribose. You're going to color those everywhere you see them. You're going to color them blue. All of them everywhere. Color all of the deoxyriboses blue. And then you're going to color the bases according to the key here, just like you did in the worksheet. So thymines are going to be orange. And so thymines are the ones that are pointy. So this is a thymine. And then guanines are purple. So guanines are cut out round, like this one here. And then adenines are green. So anytime you color a thymine, you're going to color an adenine next to it. So orange will always go with green, like this. And then purple was guanine, so the cytosine here is yellow. And I don't have yellow, so remember, if you don't have a color, click on your palette and grab it. And then it'll become part of your choices. And this one's going to be yellow. So you're going to color the rest of your DNA just like that, all of the bases you see here. Also at the bottom, it wants you to color the strands. The old one is red and the new one is green. So let's do red. So the old strand is the one on top here. So this one's old. Doing a terrible job of coloring this, but whatever. So the old one, including its bases, so each of these are all original DNA, so you're going to color all of that, and its base is red. And then the one underneath it, that's the new strand, and that's going to be green. And I think I'm going to get just a slightly darker green to distinguish from the green above. So the bottom part here is the, oh, I didn't get a dark green. The bottom part here is the old, or sorry, new is green down here. And I'm sure you'll do a much better job when you get to the coloring. And you do the same thing with the other one below. So old is red, and that would be the bottom one this time. And then new is green, which is the top one. So this one's green, and then the bottom one is red. And that's just showing you semi-conservative, half old, half new. And then you answer these questions. So each new cell must have an exact copy of the parent cell blank. The process of copying is called what? DNA replication is semi-blank because it's half old and half new. And what are the three parts to a nucleotide, which you see here? So what's the pink, what's the blue, and then what's this little flag called? And so you're going to put those on the blank there. And then you're done and you can turn this in.